again, it doesn't work, so if I would know about it earlier, I would have erased the animations, but now you have to see me run back and forth to the podium. So my apologies. Let's, before start with the paper, let's go over the two last slides from the protein structure and function chapter. And with these two slides, we will finish this chapter, where we will go very briefly on two additional methods that exist in the lab for characterizing structurally proteins, forgetting the protein structure. And the first is what is called the X-ray crystallography, which actually is the method that is used in the paper. And in this method, we first have to produce a crystal, which is a highly ordered array of the protein molecules, where they have to be in the same orientation in relation to one another. You have several millions molecules in the same orientation in order to get a strong enough signal in this method. And then you just pass an X-ray beam through the crystal and read the scatter that forms from the other side. And now the mathematics that take this scatter and transform it back to atoms, coordinates in space of atoms, actually exist for several decades already. And really the bottleneck of this method, the reason that we don't have a crystal structure for each and every protein in the nature, is really this step, the crystallization itself, where you have to play with almost any parameter you can to create such a crystal, And this is, by the way, the reason for the Nobel Prize that Ada Yonat from the Weizmann Institute received, because while other people still having trouble crystallize smaller proteins, relatively small proteins, she crystallized the ribosome, the protein that synthesized other proteins, and it's a huge protein, and it has some RNA molecules embedded in it, and it's quite difficult, and she crystallized it. And then afterwards, the next two steps are relatively known already. And another method to get the coordinates in space of the atoms in a protein is what is called the nuclear magnetic resonance. And in this method, protein solution is placed into a magnet, and different radio frequencies are activated, and one measures the energy that is discharged from the atoms while activating with these radio frequencies. And now looking at this energy pattern for a single molecule, one can determine what is the relation, or what are the correct relations between the atoms in the original molecule in the solution. And this method is effective for proteins up to 20 kilodaltons in size. So it's relatively small, but still easier to get maybe from the crystal in the crystallography. Okay, and with that, we finished the chapter of protein structure and function. So first of all, for next time, please answer the question page for this chapter. It's on the website, and I will also send it by email right after the class. And now, let's go over the paper, the NMDA receptor paper. Okay, so now we only have about an hour, maybe a bit more than that. So it will be somewhere between a frontal explanation of the paper and a discussion. We will place ourselves somewhere between these two edges on the scale. So I will ask some questions, and you can answer, and you should answer, and then in some part I will just give the long monologues. Okay, so first of all, the NMDA receptor, which is the center of this paper, is the receptor that stands in the center of synaptic plasticity. And this is, today, the idea is that this process, synaptic plasticity, is the one that's responsible for learning and memory. It's sort of the cellular mechanism for learning and memory, the most accepted explanation for learning and memory. And this paper gives the crystallography structure of this receptor. And that's why it's so important. It's in Nature and so on. And so for your warm-up, let's start with some questions about general information about this paper. So first, this receptor is called the NMDA receptor. So what is this molecule, the NMDA, that the receptor is named after? The agonist which? The agonist which activated, correct. Its ligand, the molecule that while binding to the receptor, it activates the receptor. Is it endogenous? Meaning, would you find an NMDA in the brains of living organisms in the nature? That's what endogenous means. No, it's not endogenous. It's a synthetic molecule. Okay, so in the nature, what activates the NMDA receptor? If it's a synthetic molecule, glutamate. So the neurotransmitter glutamate, which is, as you already know, an amino acid, activates the NMDA receptor. And artificially in the lab, while doing experiments or even injecting it into the animal, if you inject NMDA, it will also activate the NMDA receptor. Okay. And let's just jump for 1 second to the ninth question. What is GluR2? So there's another group of receptors that are activated only by glutamate and not by NMDA. Okay, so I would say it like that. Glutamate is the ligand for NMDA receptors and for non-NMDA GluR2 receptors. And only the NMDA receptors are activated also by NMDA, the synthetic molecule. Okay, is NMDA receptor, is it ionotropic or metabotropic? And what each one means? Let's start here, sorry, yes?
I think ionotropic.
Which is?
Directly over, it is a channel
right.
of ions and directly open channel of ions, not started processes.
Right, so NMDA receptor is an ionotropic receptor because it's a channel. So when a ligand binds to an ionotropic receptor, it activates an opening of a channel. Most usually it will be the channel itself. And then you have passes of ions, and the membrane potential starts to fluctuate to here and there. And that's a mediate response, and that's a short-range response. So that's what happens when an ionotropic is activated. What's metabotropic? Yes?
Yes, like decouple the receptor. Basically, if there is a second messenger that is activated, there's a chain of reactions. Okay. That finally can get even insight to the genes and
Absolutely. So, okay, everything's correct. So first of all, okay, so let's start it with the same way we did for the ionotropic. When a ligand binds to a metabotropic receptor, then some cascade of events is initiated, and then through really a series of events that involves secondary messengers, and we will meet these these terms later on in the course. But okay, so there's some cascade of events, and eventually there's a difference in gene expression, in synthesis of new proteins, and it's a long-range effect. The effect that is induced by activation of the metabotropic receptor. Okay. And NMDA, as we said, NMDA receptor is an ionotropic receptor. What makes it open, the receptor? Right, so you have to have glutamate, which is let's call it the main ligand. But you also have to have what is called a co-ligand. You also have to have another amino acid, another neurotransmitter, glycine, not sure about pronunciation. So you have two conditions. No conditions. You have to, and depolarization. So you have to have three things: glycine, glutamate, and depolarization, in order for the channel to be effectively open or effectively activated. Okay, so when it opens, what ions enter through what ions pass through the NMDA receptor? Calcium. Is it specific to calcium? It's actually a general cation channel. It can also, in principle, and sometimes it can also sometimes pass also sodiums and so on, but mainly passes calcium ions. Okay. Is it deactivated rapidly or slowly in relation to other receptors? Slowly, relatively slowly. And you know, what is the mechanism for the slow deactivation? Okay. Inside the cell, or what? On the receptor? Calcium does many things, but not on the receptor. And now we ask about the slow deactivation. Meaning, by slow deactivation, I mean that the channel has opened, and now what keeps it open for several actually tens of milliseconds? It can be open up to a full second, which is quite slow for receptors. And now, what keeps it open? So actually, the answer for the NMDA receptor before this paper was not really known. But if we can learn something from the GluR2 lesson, then in GluR2, the mechanism of slow activation is by this amino acid with the aromatic ring, the tyrosine, and we will see it later on. But just already to link it to what we will see later on, it's a slow deactivating receptor, and it's because of an aromatic ring structure in a certain position. Okay, is NMDA receptor heteromeric and homomeric? First, what does heteromeric mean? Right, it's built of two different units, and the hetero means different in opposed to homomeric, which is built from two identical units. Okay, NMDA receptor, is it hetero or homo? Heterodimer, actually, right? It's heteromer or heterodimer. It is built of two different unidentical subunits. 7. What is the research question of the paper, or the main one? I mean, maybe some, well, I guess there are several answers for this question, but what is the main research question in this paper? What do you think?
You want to study the structure of this NR1 and NR2, this combination of them, and this implication to physiology of the activity of this.
Okay, that's right, and it's also kind of emerged from the title itself. So you have subunit arrangements, meaning the main idea or the first idea was to get the crystallography, the exact structure in nature and function of NMDA receptors. Well, the main thing here is, the way I see it, is the structure itself. And then the question arises, what are the electrophysiology tests are here for? Why do you have the electrophysiology test and also the aggregation assays and other functional or more functional than a structure assays? And the main thing that coupled these two kinds of experiments in a single paper, again, the way I see it, is that, well, in crystallography, let's ask several guiding questions. Was the protein crystallized while binding to its ligand? No, actually no. It was crystallized as it is without its ligands. Was the whole protein crystallized? No, just the exoplasmic units. You had no intermembranal units in the domains in the crystal structure. Are the conditions for the crystal the same as the physiological conditions? Definitely not. As I said, you change as much as you can all parameters until you get a crystal, and then you're happy with yourself, and you don't really care if it's a physiological condition or not. And now, so the structure you get is in doubt. You don't really know if it's the right conformation of the protein in nature, because indeed several different conditions will change dramatically the structure of a protein. And now, in order to see if the model is correct or relevant to the biology, you check it as any other model to decide if it's a good model. You design experiments by the model, try to predict the results, the outcome, and if you're correct, then the model is good. This is a good model in science, right? So the same rule, the same principle goes here.
So this is in vivo, live rats, for example?
Here, I don't think they used rats. They used primary or other cell lines. It's still in vivo. So we will see they design experiments, and then They predict by the model. They see what will be the outcome, and then they present us the results in which they were correct, and hence the model is good.
What kind of proteins implement electrophysiological tests?
What kind of protein is, again, sorry?
So they solve the protein or is the domain, with the intracellular domain?
No, they crystallize only this to.
Okay, but
Yes.
I
do, of course. Well, if you each unit by itself is not functional, and only the extracellular units are not functional in the sense that they will not open any gate, because the gate is inside the membrane, right? You have to have the whole protein to be functional. Functional meaning giving rise to a current of calcium.
So they use several different methods to try to understand more deeply the whole structure.
Right. It's not only more deeply. It's to validate the structure they have in their hands. It's not only to go more deeply. If you go more deeply, then put this aside, the structure, or well, you can do much more things to go more deeply. And actually, there is no behavioral outcome of this receptor in this paper and so on. All experiments are designed to validate the structure they have.
So in the
Really.
Electrophysiological test,
Yes.
they use what state of protein?
The whole protein, the whole functional form of the protein.
No, a membrane.
There is a membrane. So just there have to be a membrane to record calcium currents, right? You have to have a place to stick an electrode in and measure one place in relation to another. You have a membrane there. They used some cell lines. You can open the paper and see what exactly the experimental design was. Okay. Yes?
So what they did was mutated the part that was responsible for gating, and then tested whether it's still responding to glutamate. But that still leaves open the question whether there's still some other mechanism, right? It's not 100% confirmed.
Other mechanism that is independent of glutamate, if I understand?
Yeah.
I'm not sure these experiments can really answer directly this question. But you know what, let's leave it maybe for a couple of slides that we'll go over in a second. And for now, just the principle that, again, to see why, or the coupling between these two different kinds of experiments, or the structure and function in this paper. So we've discussed this issue. And now, okay, so what glue are? We have already met it. What is the basic operational mechanism? We actually also already described it, but I'll describe it just very briefly again. So these globular domains, the exoplasmic domains of NMDA receptor, binds each one to a different ligand. And then this Pac-Man, maybe the lower jaw of the Pac-Man is closed, pulling with it the upper edge of the intramembranal domain, and a gate is opened, and ions can pass through this gate. That's the poetic operational mechanism. Okay. So the first issue that they deal with is the interaction between the two heterodomains, between the two different units. So there's an area of interface between the two. This is a side view, and this is a flip it 90 degrees. This is a top view. Thank you. Okay, and say they divide the interface to three sites of interactions. What are the interactions that are found in this interface between the two domains? What interactions would you find there? So Solbridge. Okay, first of all, hydrogen bonds, and this is like in mathematical courses. If a lecturer already take the effort to ask a question, then the answer will most usually be 0 or infinity. So now hydrogen bond is the correct answer for 99% of the questions. Okay, so besides hydrogen bonds, salt bridge. What is a salt bridge? It's a combination of two different interactions we've learned actually. The first is a hydrogen bond, and the second is electrostatic interactions. How could that be? So for instance, the interaction between a glutamic acid and lysine. So in this spot, the hydrogen will be hydrogen bonded to the oxygen right here, for the reasons we've discussed in previous classes. And also this group, the amide, has a delta positive charge. The carbonyl has a negative delta charge, and the two will electrostatically interact. And these two, I mean, this species of the combined interaction is called the Sol bridge.
Is it in the same molecules?
It's the interaction so one amino acid is from the left domain of the left unit, and the lysine to the purpose of the discussion is from the right domain.
These two have both bonding, too? The hydrogen bonding and the electrostatic?
So here on the interface you find also hydrogen bonds and salt bridges. Right?
Yeah, but you say salt bridge is both. But it's both on the same two molecules that...
No, so some of the interactions you'll find in this interface are pure hydrogen bonds, and some of them are hydrogen bonds having also electrostatic properties, and hence are salt bridges. Okay? Yep. Okay. Now let's see how well the spatial overlap is between NMDA receptor and its close relative, the GluR2, the non-NMDA receptor. So in the upper domain, in the upper subdomain of the globular domain, you have a fairly well spatial overlap, and the overlap is less good in the lower compartment of the domain, where actually the conformational change occurs, right? This is the jaw that flips up and down, and this is where, I mean, this is what we would predict, and also this is what we have, what we get. The spatial overlap is less good, and we can, using a crystallography structure, we can even measure how good or bad is it. So, for instance, the distance between the linkers, which are the sites that link these domains to the intermembranal domains, differ in 6 angstrom between GluR2 and NMDA receptor. You don't have to remember by heart this detail. It's just to give you a feeling of what we can get from crystallography structure. Yes.
Is this pseudo-soil twofold axis they are talking about? Because I don't understand what it means.
So we won't enter the pseudo. What's the second? Twofold axis. Never. It's there's some issues of symmetry in crystallography. So we just don't want. I don't really know that these things really good enough maybe to teach it. And also we're not interested in that for the reason I've discussed and because of the time constraint. Okay, now a question that was standing for several decades actually is how come or why? What's the mechanism? What's the reason for the fact that NMDA receptor is activated by NMDA and not other non-NMDA receptors? Because both of them are glutamate receptors, and they are very similar in structure, and they deliver the same currents and so on. So what's the reason for the first to be indifferent for NMDA and the other to be responsive to it? And we have an answer. This paper gives you the answer. So what is the answer? Right.
NMDA was less one soul bridge, and then the NMDA is speaking to the something.
Right. So again, we will limit the discussion only to the first reason you gave, which is the shorter amino acid in the active site or the binding site of the NMDA receptor. And the story goes like this. So we are looking right now at the ligand binding site of NMDA receptor, and here of NMDA receptor overlapped. We see the same structure at the same figure. It might be a bit inconvenient, but that's what they give you of NMDA receptor and GluR2 receptor. Let's start with the upper panel. So this is the active site of the ligand binding site of NMDA receptor, and you see some amino acids in it. Note the D731. This is aspartate. D is the one-letter annotation for aspartate, and this is NMDA molecule. What you see here? Sorry, it's the it's the glutamate receptor. It's the endogenous ligand. Okay, and you see it fit inside the binding site of NMDA receptor while interacting via a water molecule right here. Okay. And now, because the interaction here is via a water molecule, you can take this water molecule out and feed inside the NMDA molecule, which is actually like the endogenous ligand, the glutamate, having another carbon right here instead of the water molecule. Again, structurally, NMDA molecule mimics the structure, the general structure of a glutamate with a water molecule here. And that's the reason why you can take this water molecule out and place inside the NMDA molecule with its additional carbon right here. This is the one that stands here instead of the water molecule. And it fits inside the ligand binding site of NMDA receptor. If you try to do the same thing in GluR2, you will not succeed, because instead of this aspartate, it's not the aromatic ring, it's the one that's beneath it, the aspartate. You see here its structure. It has no aromatic ring, but instead of the aspartate in the NMDA receptor, you find in the same position a glutamate. And now don't be confused with the ligand, with the endogenous neurotransmitter itself, glutamate. There's another glutamate in this story, the amino acid from the polypeptide chain in the GluR2 channel. So in this time, in the position where the aspartate stood in the NMDA receptor, you find glutamate, which is longer than aspartate in one carbon. You see it here. The chain has one carbon more than aspartate. And this is this chain here, the red one. And in this case, when you try to fit in, to really stick in the NMDA molecule, there will be a collision of electron clouds between this carbon, which is the yellow sphere here, and the carbonyl groups of the glutamate. The glutamate here means the glutamate, the amino acid from the backbone of which is part of the ligand binding site of the protein. And that's the reason why NMDA molecule cannot fit in to the non-NMDA receptors and can fit in to the NMDA receptors. Again, this time we'll begin by introducing the players. Aspartate is the amino acid in the NMDA receptor. It's an amino acid, yeah. Right. It's an amino acid in NMDA receptor in its ligand site, in its binding site. Sorry, it's shorter in one carbon than glutamate, the amino acid in the non-NMDA active site. And again, don't be confused. Glutamate also plays two roles in this story. It's the ligand, the neurotransmitter, and it's also a structural amino acid in the binding site of the non-NMDA receptor. And NMDA molecule, which mimics the structure of a glutamate with an additional carbon here. And again, the active site of NMDA receptors interact with NMDA plus a water molecule. This situation activates the receptor. If you take out the water molecule, you can structurally fit in the NMDA molecule. Again, the outcome is activation of the receptor.
Molecule is not part of the receptor. It's an alternative molecule, like the glutamate.
Like the?
Glutamate.
Like the glutamate, absolutely. Yeah, it's not a steady part of the protein. It binds to it with the glutamate.
So you need to have the water molecule for the receptor to be activated?
With glutamate, not with NMDA. Yes, yeah, absolutely. Again, it's a question of stabilization. I guess some of the cases the glutamate will activate for a tiny fraction of time the receptor. It's only a question of efficiency. Yeah, right. So the most efficient form is with the water molecule. Sorry. Yes?
And they release the water molecule?
Again, the NMDA?
NMDA molecule, when it activates the NMDA receptor.
Without the water molecule.
So it releases the water molecule?
It's not active. Again, the water molecule binds into it together with glutamate. So when NMDA is present in the system, you just fit into the active site without this water molecule.
Where
right?
This molecule
the environment is filled with millions of water molecules. It joins its rest of the family. And so, okay, and because this is the aspartate from the NMDA receptor, which is shorter in one carbon than the glutamate in the non-NMDA receptor. And if you try for that reason exactly, the length of the glutamate, if you try to stick in this case the NMDA molecule, there will be a collision in electron clouds between the additional carbon in the NMDA and the edge of the glutamate. Chop the glutamate in one carbon, and you could succeed in fitting inside the binding site the NMDA molecule. So that's sort of what nature did. He produces a mutation, I don't know how or when or why, but it produces a mutation that switched glutamate to aspartate, or actually in the inverse order. We don't really know. Question? Okay, so again, this was a major question, at least in laboratories. We didn't know why it works so well for some receptors and not for others. So this is the question, the answer. Okay. So, and let's move on to some of the functional assays they did. Not so fast. Okay, so we have a model, and we want to check if it's valid. So we design experiments using this that are based on this model, and we do these experiments and check if we can predict correctly the outcome. Okay, so the first thing they did, what's described here in this set of experiments? What mutations that the researchers introduced to the protein? Right. So switching between any amino acid and cysteine is unique in what sense? What's so special in cysteine? The SS binding, what we call the cross-linked via cysteine bond or something similar. So two cysteines that are close together in oxidizing environment or oxidizing enough will be covalently linked. This is something that happens only for cysteines. In many papers, you see mutations, switching mutations to cysteines. And this is a nomenclature for mutations. So, yes?
Why is it important, the binding between cysteines?
For this?
No, why is it so common? Why is this the property of the amino acid is?
Why only cysteine covalently linked to itself?
Why is it important?
Why to use it here?
Yeah.
So,
you said
okay.
In many studies.
Okay, so I'll give the reason here, and I think it will imply the reasons for other papers. But one more second. So the nomenclature around mutations that are done in proteins, and in this protein they did the L amino acid. Can't remember what it is right now. Was switched in this position. L in this position was switched to cysteine. E in this position was switched to cysteine. Something switched to cysteine. Something switched to cysteine. Okay, and what's the idea here? So you take amino acid, you have a structural model. Without it, you couldn't say that this amino acid from the purple domain is a neighbor, spatially neighbor of the green amino acid of another amino acid from a different unit. Now the structure says that the two are very closely, just close in space, and then if you switch the two of them to cysteines and give them the right conditions, they will covalently link. This will not happen if you will do point mutations in two sites that are far apart. For instance, this one and this one. Mutate these amino acids to cysteine, and the same for this amino acid. Give as much oxygen as you want, and you would not get a covalent link. So you predict that doing this, you will get a covalent link between the two units, and that they will be aggregated stably and will even be found together in a gel. So 1 second before the gel. Again, the switching of any amino acid in these two positions to cysteine is clear, right? Is it clear? That's the meaning. Okay. Okay, so now you run this thing on a gel in oxidizing environment, the environment in which cysteine bonds exist and are stabilized, and in what is called reducing environment. Just read, a few sentences about that in Wikipedia or whatever, what oxidizing environment and reducing environment means. And in the environment that holds these interactions, so we have several controls here. Never mind the first one. Wild type. If you without any mutations, the two will not bind to one another, and will hence each one will run on the gel separately, and we will find a product lower on the gel, right? Because lower means smaller size protein. Even if you mutate just one unit and not the other, it's not enough to produce the link, right? You have to have two cysteines, one in front of the other, to get the link. So any single mutation wouldn't do. And if you do double mutation, well, most of the proteins might be unimpaired, or you couldn't induce a covalent link in them, but some of them, and it's a clear product, have aggregated because of these cysteines. And the right-hand gel is a control for the fact that it is the reason is the cysteine bonds, because one thing we know about them is that they are destroyed in reducing environment. Okay, let's leave this and this one aside. Yeah, these products are when the two run separately. When the two units just run separately on the gel.
They engineer them separately? Enter them together into...
They engineer them and then isolate them, and then do some purification procedures, and then run them on the gel, and they run separately. Okay.
The only thing they check is that the 2 points of the amino acid L and C are closer together.
As the structure would predict, yes.
But everything else can be different.
Right here in this experiment, yes. Basically, you're right. Okay, and now there's also a functional. I'm not sure why to do. Okay, never mind. It's a dead assay. I mean, this protein might not be functional here. Actually, it's not functional because it's a denatured gel. Okay, so it destroys the tertiary and secondary structures of the protein. That's the idea behind it. So in some conditions you will, again, gels destroy any interactions, put aside covalent interactions. And this is why only in the double mutant you get this product and not in the other lanes. And functionally, they also did electrophysiology of the wild type, the mutant of one domain, and the mutant of the second domain, and the double mutant. Each time they record current influx in oxy-reducing environment and oxidizing environment. So this is wild type. Why is there a difference? What do you think? Why is there a difference between the two environments? It's a tricky question.
What do we see?
What do we see? So what do we see here? You're right. What do we see here? So that's so the units are nanoampere. It's current for calcium because that's the ions that pass through this channel, right? And the current that goes downwards means positive ions enters the cell. So this is just a convention. And this is what we see, the calcium currents that are produced by this channel. Okay, so again, wild type. No new cysteine bonds. Why would the environment make a difference? Right, maybe there's some endogenous cysteine bonds in the protein that we have not introduced. Some of these amino acids here might be, and probably are, cysteines. So destroying them would change the function of the protein. Okay? So this is why we have this delta in each experiment between the reducing and oxidizing environments. Single mutants don't do such a difference, and the double mutant, you see a big difference when, like, pressing on the induction of the mutation. So when the two cysteines are the cysteine interactions are stabilized in the environment they like to live in, you see less efficiency in delivering currents into the cell, in in transferring ions of calcium into the cell. Sorry? It's well, you have to have cells here. I mean, you record the protein. It's not a. It's I think it's primary. It's red. The mutated protein is introduced in vivo in some way. I can't. So you give me the answer. If it's a cell line, what? So it's okay. So it's frog. It's frog. The protein by itself cannot deliver any currents. You can't measure any currents if the protein is isolated in a test tube. Currents can only be recorded when you have a difference between between inside and outside of somewhere, of some membrane. Okay. So now, never mind if it goes up and down. It has some functional outcome, desystain mutations. Okay, as I said at the beginning, the GluR2 receptor, to which we already have a structure, a crystallography structure. So when you look at it, you find a tyrosine in a specific site. And we also know that this tyrosine plays a role in the slow deactivation of the GluR2. When we look at NMDA receptor, we find the same amino acid, tyrosine, in relatively the same site. And the question arises whether also in the NMDA receptor, the tyrosine plays the same role. Does it have an effect on the slow activation of the receptor? And the answer is just yes, because when we do mutations of this tyrosine, here in the schematic model, to an amino acid with no aromatic ring, you see faster deactivation. These two mutations I've just said are the blue and black, and the wild type is red. Wild type means that you have a tyrosine in this site. And I think the more interesting thing is the mutation to tryptophan, which has a maybe bigger to two aromatic rings. Anyway, the mutation to tryptophan is kind of a gain of function, not a loss of function mutation, in the sense that you have this is the green curve here. You get even a slower deactivation than is the case in the wild type. It's cool just because destroying things in cells is not that hard, but trying to do some gain of function mutation, this is kind of I think it's more an exception, and that's why I think it's more interesting. Okay, and you can fit this curve and check the tau, and you can see how they did it in the paper. This is the quantification of slow and fast deactivation. So yes, for NMDA receptor, the tryptophan here is essential, because without it, the tyrosine is essential because without it, the channel will be open for only a tiny fraction of time, and with it, it has a slow activation, which is thought to play an essential role in plasticity, in synaptic plasticity.
NMDA is only in the brain?
Would NMDA be found in other tissues? I'm not sure. We know it's in the brain, or almost anywhere in the brain. The most prominent experiments were done in hippocampus. I'm not sure. I don't think so, but I'm not really sure. Okay, and that's that about the paper. Philosophical word to conclude, to end this paper. So as I said in the beginning of the course, I mean, for some researchers, the question of whether a learning and memory will be formed. So some researchers ask this question like this: whether would you be able to diagonalize the connection matrix, right, in neuronal networks from Heimson-Polinsky? This is the right question to ask in order to understand how memory is formed. For other researchers, whether a memory will be formed or not is dependent on whether you'll find a specific amino acid with the aromatic ring in it in a specific site on the protein. So this is so you see it's like two languages. How to get between the two, I have no idea, but it's really two distinct languages, and that's good. I mean, it's okay. It's part of the game. And that's it. Any questions about the paper? because you don't want to delay the break. If you have any questions about it afterwards, then you're also welcome to ask. So let's meet after the break.
שאלה קטנה
כן, מלך.
זאת אומרת שה-NMDA לא נמצא בטבע?
לא נמצא בטבע.
אז מה בכלל המשמעות שלו?
המשמעות זה כן.
יש לך את הגלוטומט שהוא הלייגנד
הוא מהכתב אותו. זה נשמע כמו שאלה אולי לא חשובה כל כך, כי באמת היא כאילו לא קורית בטבע, אבל שנים של ניסויים, טונות של ניסויים, שבהם אתה מהכתב ואתה לומד את התעלה.
אבל למה לא לעשות את הניסויים עם גלוטומט?
אוקיי, מכל מיני סיבות, זה פחות יציב, NMDA אתה יכול לייצר את זה ביותר, אתה מייצר את זה ממש בקלות, זה פשוט חומר אורגני, אוקיי.
אני לא מבין את התהליך בכלל שהביא, זאת אומרת, יש לך רצפטור, איך ייצרת
וגם
חלבון שמתאים
אמפירית, אני חושב, מאיפה זה הגיע בהתחלה, אני לא בטוח. בדרך כלל הדברים האלו מגיעים מרעלים, שאז אתה מוצא משהו שדווקא דופק, ואז אתה מצליח דרך הרעל לבודד איזה משפחה. הרי אתה בכלל, יש לך מלא חלבונים בתא, חלקם שונים באיזה סוכר שנמצא עליהם. אנחנו מגדירים אחרי זה, ואנחנו קוראים לזה משה ולזה איציק. אבל קורה לפעמים פלופ של הגורל, שבהם הכנסת, כאילו, אז או שבאמת רעלים וכאלו, או שבמעבדה הכניסו את החומר הזה והמשיכו לרשום, ופתאום, זאת אומרת, לאו דווקא הזרם הכללי של הקלציום ירד או עלה, פשוט השתנה פתאום. נגיד, הוא מגיע לאותו פיקר כשהוא עושה הרבה יותר slow deactivation. אז הרסת איזה סאב-פופוליישן של רספטורס, שבשלב הזה עוד קשה לך להגיד מי הם. ואז כאילו ממשיכים ורואים שאתה מבודד את הסאב-פופוליישן הזה. אמפירית, למה השתמשו ב-NMDA? אני חושב שזה פשוט יותר פשוט, יותר הדיר. כן?
זה אותי בלבל שקוראים לו NMDA, אז ברור שהוא משוייך ל-NMDA רספטור.
נכון, אבל זהו, הרצפטור הוא לא סינתטי כמובן, הוא נמצא בטבע. עכשיו הוא שונה, יש כל מיני גלוטמט רספטורס. יש המון, יש גם גלו-R2, אני מניח שמסתכל טוב מספיק, מתחת למיקרוסקופ טוב מספיק, תראה שגם הם נבדלים ביניהם.
אז כאילו גם לגלו-R2 נגיד אתה יכול למצוא משהו מסונטז
בקטע הזה. שוב, אני מניח. משהו מסונטז שיתאים רק לו ולא לחלבון אחר. אני מניח, אני לא יודע. פשוט זה כזה נתון אמפירי שבאמת תמיד הכניסו, כן, הכניסו NMDA וראו שמשפחה מסוימת של רצפטורים מופעלת ולא אחרת. עכשיו גם, כן. כאילו באמת הרלוונטיות הביולוגית כאן אני לא יודע להגיד, אבל כל כך הרבה ניסויים נעשו עם NMDA שאפילו בשביל הקהילה זה חשוב, התשובה הזאת. כאילו התשובה המורכבת ללמה NMDA ולא גלוטמט מתאים לפה ולא לשם. כן. זה בערך תשובה?
כן, כן, כן.
אוקיי.
ועוד שאלה, אני לא זוכר מה אמרת בתחילת הזה, המאמרים הם למבחן? לא?
לא, אני אגיד שוב אחרי זה. המאמרים הם לכיף. סבבה.
לא, אבל אני מניח
מאמרים הם בשביל הנשמה. סתם, בסדר, כן. לא, אני אגיד.
כאילו, המאמר עצמו לא, אבל כל מה שיש בו
זהו.
זה...
מה?
לא, ברור.
הם לא, אבל אז כאילו בגלל זה אולי גם שמנו את ה-20% של המאמרים, שכאילו תופיעו לכיתה ותדעו על מה המאמר ומשהו כזה. כן.
כי היה לי מאוד קשה לקרוא,
סבבה, נכון, אבל כאילו הרעיון... זהו, אני מקווה שזה לא לגמרי גם זה, כאילו הרעיון בכל זאת היה שהשיעורים האחרונים יחסית מסבירים כמה שאפשר. בסדר, ברור, מלא גאפס.
אני מניח שאם היית מסביר את זה ולא הייתי קורא לפני זה, אז בכלל לא היה לי מושג למה לדבר. אז ככה לפחות זה התקשר לי.
כן, יש פה מלא, נכון, מלא נומנקלטורה מעצבנת כזה, כן? סבבה. תזכיר לי את השם? אורן, סבבה, נכון. gene when it's already a protein, you mean functional switch it
No, I
on?
mean like when you express it in whatever your whatever like the oocyte, in this
Yeah.
example, like you just do an easy manipulation to switch
The words easy and simple are kind of, you hurt me by saying it, but it's not really easy or simple. When it's clean, when it's working, it's working. That I agree, but it sometimes takes several years to get a clean system in it. Because if you know, if you want to switch a gene on, maybe you add some thing that it binds to its promoter, some regulatory element that before the gene in the genome, and which will open the genome in this specific region. But when you do so, you have much more responses that you haven't predicted, you haven't thought that would happen, and then you have to start, abolish them and fight with the artifacts.
right, so we haven't gotten it. Okay.
And so it's not easy, but when it works, it works.
That's because the hard part of that paper is the crystallography, no?
Over here,
The actual crystallization.
well, in the sense that it was the methodological bottleneck. People tried for several, I'm not sure how long, but they tried for many years to crystallize this protein because it's from the 50s. It has its name and people know that it's important for them, or think that it's important, and then people tried to crystallize it in vain. And electrophysiology, already existed.
Right.
So yeah, it was the bottleneck. I'm not sure about it was the stuff of the bad part, I would say. Things.
מה כתב המאמרים האלה? אני מיואש מהם.
מה בסוף מהנדרה זה הראשון, נכון? זה לא?
כן.
אוקיי, אז
לא,
מה
היה
מיואש?
במקום אחר, אני מפיל עליך גם
לא,
במקום אחר.
בסדר. מה זאת אומרת מיואש?
סתם, לא יודע, כאילו זה, בוא'נה, אתה לא מבין כלום.
לא, אז תגיד באמת, טוב, אני אגיד לכם, תראה, הם לא למבחן.
כן,
לא למבחן. אז תראה, אתה לא נדרש באמת לדעת, כאילו.
כאילו, אני לא יודע איפה עובר, כאילו, איפה עובר הגבול בין להבין ברמה כללית לבין פרטים.
אז זו שאלה חשובה שאתה אומר, תשאל אותה בעיקר על החומר, כי הוא החומר עצמו למבחן,
לא, אבל גם פה זה מצוין. אנחנו סטודנטים, אנחנו נבין ציונים.
אז פה הציון, זה בסדר גמור, אני אגע, אז פה הציון הוא כמו שהיה עכשיו מבחינתי. אני מדבר עליך ספציפית. זה שאתה מראה, שאתה יודע מי הרצפטור, מה הניסויים שנעשו, איך הם מתקשרים אחד לשני, ואז מיליון פרטים, כמו למה ציסטין, למה שם, זה התפקיד שלי, לא שלך. באמת הרעיון פה הוא לא...
לא, אבל זה קשה להבין משהו אם ייתן לך את כל הפרטים, כאילו,
סבבה.
אתה מקבל, כאילו, אתה לא יודע להבדיל בעיקר לטפלים, אתה לא יודע מה עושה הציסטין, אתה לא יודע להבדיל בעיקר לטפל, אתה מבין? מה היה העיקרון פה?
אז העיקר פה הוא עקרוני, העיקר פה הוא לנסות להבין את המאמר ולהבין כמה שתצליח בתוך המאמר. זה ממש עקרון פה, בגלל זה לא במבחן?
כן, זה לא חומר שעוד לא למדנו בכלל על קולטנים ועל...
נכון, בסדר, וגם אחרי שתלמד, תחזור למאמר לאו דווקא תבין הכל, זה לא, זה באמת לא נורא
לא,
כבד.
אני כאילו
ישבתי,
אמרתי למישהו, הוא עושה לי לדלג את החלק הזה של האקסטרופיזיולוגיה, כי אין מצב שדרשו לכם להבין דבר כזה לפני שעשיתם קורס ביולוגיה מולקולרית.
אז על הכיף.
כאילו ככה הוא אמר לי, כאילו לא...
אז אני אומר פעם, אני לא
ניסיתי להבין,
דורש. מצד שני, זה קורס לתואר שני, כן? אז קורה שהם דורשים משהו שלא למדת, זה לא...
בסדר, מקובל.
לא, אז אני אומר אותם, זה לא במבחן. הרעיון הוא רק ההתנסות, זאת אומרת, הרעיון הוא לא שלא תבין כלום, אני לא צריך להגזים. אני בטוח שלא הבנת כלום. בסדר, העניין הוא קצת, הרעיון הוא קצת לשחות בבניין הזה, במבנה הזה, להסתכל עליו, לראות אותו וזה, ולראות, כאילו קשה לי לנסח את המטרה, אבל המטרה היא בדיוק מה שהיה הרגע, מבחינתי, וזה לא מבחן.
לא, חייב את זה, כאילו, זה בסוף את הקטע, כאילו, אני נניח, אני בא לקרוא את החלק השני, אתה אומר שהם עשו איזשהו תוקף חיצוני, איך שקוראים לזה, לא יודע אם קוראים לזה גם ביולוגיה ככה, כאילו, הם בדקו משהו, הם עושים לבדוק את זה גם בטבע ככה. אז כאילו, אני באופן טבעי, אני אומר, הם בדקו את זה על איזשהו תא חי, כאילו, יעשו איזה ליבוי ויזריקו משהו לעכבר חי, או זה החיה או לטבע.
אז השאלה היא מה אתה רוצה לבדוק,
כאילו, אני פשוט לא מבין מה הקשר, כאילו, כשאני לא מבין את הטיסים הזה, אני פשוט לא מבין מה הקשר בין זה לבין תוקף חיצוני.
מעולה, עכשיו אתה מבין יותר אבל? בוא
עכשיו אני מבין.
סבבה. אז זה שבבית התכוננת לזה, זה כדי שעכשיו תבין גם, ואפשר עוד לדבר על זה מבחינת התוכן, אז תראה, אנשים עוברים את כל הטרחה של להזריק לעכבר, בדרך כלל שמדובר באיזשהו ניסוי שגם כולל התנהגות. אם מדובר על כמה איונים עוברים כשהחלבון נפתח או נסגר, לא הייתי הולך לעכבר, כאילו זה קשה. עכברים מתים כשאתה מזריק להם, הם יקרים, זה כואב להם. ביחס נגיד להזריק, לא יודע, לגדל לתאים או לצפרדעים או לדברים כאלו.
לצפרדעים אפשר להזריק חופשי?
תראה, יונקים זה, כן, כאילו אתה יכול להזריק חופשי לעכבר. לא חושב, זה כבר חיות כאילו? מה אתה אומר? צפרדעים חיות? אני לא יודע איך לעשות את זה.
זה עדיין מוקלט כאן?
מוקלט. חוזר שלום.
זה באתר?
זה יהיה תוך איזה יומיים כזה. זה בעצם תמיד משדר, אבל אז חזי, מישהו שנתיים על זה, אז הוא עורך את זה, ואז זה נכנס לעניין. תוך איזה יומיים. אני יכול לשלוח להם כל פעם שאני מפרסם.
רגע, כלומר, זה ששיעורים שעברו כבר מופיעים שם?
כן, כן, בלינק ששלחתי. כן, כן.
צריך להציג את זה מישהו שיפתח מייל עם ה-SSC?
לא הבנתי. זה ביוטיוב חופשי.
מישהו שיפתח מייל, שיפתח במיילים, אז לא יודע מה חשוב ומה לא.
זה אני אחר. כן, אתה יכול להגיד את זה. they are. So the idea behind it, when we're discussing the papers in class, is to first maybe to show you that the material we've learned is properly implemented while reading papers. The last classes we sort of tried them to be gradually built towards this paper. Most of the terms have been mentioned in previous classes and so on, but it's not a part of the final exam, partly because it's a heavy paper, I know. It has almost any methods that exist in biology, electrophysiology, aggregation, and the way the discussion went right now, I think it's effective, and we'll proceed in this level of discussion, in which you read the paper at home, know very well what is the research question and what were the methods that used, maybe read a bit them outside the paper even, about how you perform them, and then how the different experiments are linked to one another to answer the research question, and that's the idea for the discussions about the papers. And also the 40% that belongs to the home assignment in the end of the semester. So the questions will be very similar to the questions we ask about papers here in class. So another thing is the kind of a practice to the final assignment, to the home assignment. But obviously it stands by its own. It's an amazing paper, and that's why we are learning it, right? So that's the main idea. Not physically, really. It's a very okay. And that's all I wanted to say. Okay, so the next chapter is a very short one. Basic molecular genetic mechanisms. Okay, so before starting it, actually I would say a word about future chapters. So the next couple of chapters are built gradually starting with the architecture of cell membranes through general discussion of intercellular signaling and ending with a specific family that plays a part in intercellular signaling. And this chapter is kind of this is where we succeeded in placing it right after the protein structure and function, and before we proceed with more deep issues of intercellular signaling. So we have to maybe put us all in the same level regarding the central dogma of gene expression. So I predict it to be a bit too obvious to students with background in biology. But for those without a background in biology, as any other chapter, there will be quite a lot of new terms and new definitions, so it's a chapter as any other. Yeah. So in this chapter, there will be some more information about DNA, right? An RNA structure, and we will describe these different steps for the central dogma. Dogma in English? Dogma of gene expression. It's parallel to chapter 4 in Godish textbook. So it's actually an overview slide for this chapter. So we'll see we'll describe the different transition steps between DNA and RNA. This transition is called transcription, and when RNA becomes a protein, it's through a process called translation, and we see some details about these two steps. And already at this point, I'd like to emphasize the different roles that RNA molecules play in this dogma. So they have a role as information storage when they are in the form of DNA and messenger RNA. They have a role in sequence interpretation as transfer RNA, tRNA, and they also have a structural role in ribosomes, which are the proteins that synthesize other proteins, in which case they are called ribosomal RNA or rRNA. And we will see these three in a couple of slides. There are also some regulatory RNA molecules which we will meet in future classes, such as microRNA. Let's put the central dogma in its spatial context in the cell. So DNA is localized in the nucleus, where it is being translated into RNA. RNA is then processed in many ways. We will see these different processes later on, and it then leaves the nucleus to the cytosol, where it is being translated to proteins. And this is locally where things happen. This is a reminder for about the DNA structure. So these are the nucleotides, right? Nucleotide 1, nucleotide, second nucleotide, and they are bonded via phosphodiester bond. Phosphate link between two oxygens to other molecules. So this is a phosphodiester bond. And together they build the nucleic acid chain of DNA. DNA has the structure of a double helix built of two strands in an anti-parallel orientation to one another. The orientation of a single strand is defined as the following. So in each strand we can kind of identify an edge with an exposed 3' edge. The 3 here comes from the convention of how to count carbons on a sugar molecule, but never mind that. One of them is on the 3' and the other has an exposed 5'. But you'll see that the nucleotides are facing a certain direction and are not, flipped back and forth along the molecule. So you have an orientation for each strand in the DNA double helix to find the two strands in parallel orientation to one another. One goes up and the other sort of goes down in its direction, from the 5 to the 3', and its adjacent parallel strand from the 3 to the 5' end. In this double helix, the sugar phosphate backbone face outwards, and the bases, remember we call these moieties bases, they protrude towards the interior of the molecule, where they interact with each other. A purine linked to a pyrimidine by a series of hydrogen bonds. Yes, so these hydrogen between the bases are hydrogen bonds. Besides the hydrogen bonds along the molecule also hydrophobic effect and van der Waals interactions stabilize the DNA double helix. But specifically, these hydrogen bonds between a purine and a pyrimidine is called after the two researchers that deciphered the structure of the DNA double helix, Watson and Crick. So these are called also Watson-Crick interactions between two bases.
The hydrogen bonds?
The hydrogen bonds between the purine and pyrimidine are Watson-Crick interactions. And they are the consequence of the shape and the size and the distance between each of the nucleotides, each of the bases in the nucleotide. In the DNA double helix, there are two grooves are formed: a major groove and a minor groove. Gumcha. A hole. Groove is like a gumcha. Okay. Explaining one word with another one, that groove is a groove. What? It's like a crevice. Any better? A crevice is a groove and a groove is a crevice. כן, גומחה זה בסדר. גומחה זה כן. Okay, so the major groove is right here where the nucleotides are, where the bases of the nucleotides are actually exposed to the environment, because right here, the long red and blue lines are the sugar phosphate backbones. And what distinguishes a specific region of the DNA double helix from another are the bases, not the backbones. And these regions are exposed right here in the major groove or the minor groove. It's where the nucleotides are exposed. And this is where proteins that interact with the DNA bind to. These are the Watson and Crick interactions. All right. So the basal form, the basal form, the most common form of DNA found in cells is the so-called B-DNA form, which is the form I've just described. Its characteristics are that it's a right-handed helix, and it makes a complete turn every 3.6 nanometer, means about every 10 nucleotides. So if you go along these curved steps, 10 steps, you will make a complete turn. Again, the characteristics for the most common form of DNA, which is called the bDNA, is that it makes a complete turn every around 10 nucleotides. If you mark this here in black, you will find it again in 10 nucleotides again in the same position. But we know that DNA can be found in other forms as well. The so-called A form, which is a more compact steel alpha helix DNA, and also we found in nature a left-handed, more elongated structure of the DNA helix, the Z form. And for many years we thought that these two structures exist only in test tubes, in laboratories, but they were found in nature already. And we think that their role is the following one. While DNA is being transcribed to RNA, you have to it's called a melting. You have to melt these two strands apart. You have to break these connections apart and take them farther from one another. And then a window is formed right here, or what is called the bubble, a transcription bubble. And doing that, you accumulate tension in both edges of the bubble, in both edges of the window. These two structures have higher tension loads. They can absorb more tension simply because of their characteristics, structural characteristics. So these two structures are formed in the two edges, thereby absorbing some of the tension that you have released while opening a transcription bubble.
So,
Just imagine, once again, just imagine a helix or a strand that is melupaf, a strand that you just twist its two edges, and in the middle of it you'll find a lot of tension, right, simply because of the twisting. And if you open in the middle of it, there will be more tension in the edges, simply because even the helix itself will become more compact.
So
So this is exactly what happens
on the same DNA?
here. On the same DNA molecule, yes. Definitely on the same DNA molecule. So they have a role in reducing the tension in the transcription bubble during the process of transcription.
And you said that in these two you have more tension on the edges, and then how you produce the tension?
I say that in these two, as they are intrinsically, there is more tension. So they sort of can absorb tension from the edges during transcription. Or in other words, when transcription happens and you open this window right here, so these two forms will be formed in the two edges. These two forms which are more compact, which have more tension. Is that good? So I'll try again. Actually, it's not, because it's really in science a novel new field, giving roles for these different forms of DNA, the A and Z forms. But again, the most common idea is that these two structures are formed in the edges of the transcription bubble because they absorb some of the tension that is released in the while opening the transcription bubble itself. Give me 1 second, please. Yeah, I have. So as we saw in the previous slide, the Watson-Crick interactions between the nucleotides are not parallel, but rather perpendicular to the axis of the molecule, and that's what makes them different from the alpha helix in the proteins. So, when we discussed about the secondary structure alpha helix that's found in proteins, which said that it has interactions parallel to the axis of the molecule. By axis of the molecule, I mean this axis. So these were the interactions between moieties in the backbone of the amino acid polychain, the amino acid chain, right? In the case of proteins. And now I tell you that we also have a DNA alpha helix, but it has a very distinct properties from that of the alpha helix found in proteins. And the major property that really makes these two structures very different from one another is that the structures here in the DNA are not parallel to this axis, but rather perpendicular, right? The Watson-Crick interactions were between nucleotides that face each other on both sides, or from both sides of the DNA double helix. And so this structure found in proteins this structure opposes forces that goes towards the sides. It will be very hard to bend this molecule here or here, but bending DNA to either side is pretty easy. There's no opposing force to this bending force, because the interactions are in this orientation. Okay. And indeed, when DNA binds to proteins, when it's being transcribed or when it's being remodeled, or when the cell is going into its cell cycle, so the DNA is indeed being bended, sometimes very sharply, in order for good recognition to be produced with some protein. For instance, the Tata box binding protein. The box is the sequence on the DNA. The protein that recognizes it has to bend the DNA in order to recognize it efficiently, as seen in the picture.
The data box is on the DNA and
Yeah.
like a binding protein.
Yes. Data box is the sequence on the DNA, and that's the protein that binds to it. So the transcription process that takes the DNA and transfer the information stored in it to RNA molecules can be divided to 3 steps: initiation, elongation, and termination. And termination by itself can also be subdivided to 3 steps. Well, in the first, the protein that's called RNA polymerase. Sorry? So now I'm talking about initiation. Sorry, not termination. My fault. So the first step of initiation is where the RNA polymerase. That's the protein that promotes linkage between two nucleic acids. Yes, sorry. Finds a transcription start site on the DNA, marked here in green, and then it opens what we call the transcription bubble. It has some melting properties, the RNA polymerase itself. It's a protein complex, so some proteins on it, or some enzymes that build it, have melting properties that can melt the 2 strands apart from one another. And then the first link between the first 2 nucleotides is formed, and that's a relatively slow process. Then we enter the elongation step, in which lots of new phosphodiester bonds are being formed between nucleic acids. And this step is really rather, during elongation, the elongation of the RNA is pretty fast, pretty rapid. RNA polymerase in this step has high efficiency in the sense that it's very rapidly attaches new nucleic acids. And when it meets its stop codon, again a specific sequence on the DNA, that's when we call this process is called termination. So when it binds to this stop codon, the RNA polymerase dissociates from the DNA template, also releases the complete RNA strand.
Codon is not term for mRNA sequence?
Also, but not only. To stop, yeah, you can also say stop. A triple codon is when RNA molecule transformed into amino acid. We'll see it in a minute. A codon is just a specific site on the DNA or RNA that codes for something. Yes? Yes.
You said that some process happens slowly and something is fast?
Yeah. So what RNA polymerase does is actually, is attaching one nucleic acid to the other via a phosphodiester bond. It does it in the last right here, this short red strand means that the two first nucleic acids were already linked by a phosphodiester bond. This link is rather small, and then the next couple of linkages are still kind of slow, and then it becomes very, very rapid, the attachment of new nucleic acids. So in elongation, it can transcribe a whole gene just in a matter of even seconds sometimes.
What I should do is to bond according to the base pairing rules. Not,
Again, sorry?
it should bind nucleotide to nucleotide by
Complementarity with the DNA.
Yes.
Yes, so of course. Okay, so the RNA is being transcribed according to the DNA template. Each nucleic acid is not randomly chosen from the environment, but rather complement or complement to the nucleic acid that's found in the DNA. So at a certain position of the RNA polymerase, in this complex you'll find the DNA-RNA hybrid, and the RNA strand here is really equivalent to this DNA strand, besides the fact that it has guanine instead of thymine and so on, but it's exactly the same sequence of nucleic acid.
And the Watson-Crick bonds don't depend on the polymerase, the RNA polymerase.
That's not the right phrasing. I mean, the Watson-Crick bonds are, their creation is promoted by the RNA polymerase.
So it's promote the Watson-Crick bond
Yes.
and the phosphoester bond.
Yeah, okay, yes, correct. Okay, and that was termination. From that second to up till when the RNA becomes a protein, it's in a, most usually in a single-stranded form. And single-stranded RNA molecules can form several structures. Here are some examples: hairpins, stem loops, pseudoknots, and as we did in protein, when we discussed secondary and tertiary structural levels, the same discussion can be make sense here for RNA single-stranded structures. So you can find secondary structures and tertiary structures. We won't do it really right now, but this is just to show that RNA molecules are not boring and they have specific structure in space and not only a primary structural level. Okay, and this figure describes the process of transcription when RNA is transcribed into protein, to a sequence of amino acids. And it also gives us in it you'll find the three different kinds or main kinds of RNA molecules playing different roles. So the first one is the messenger RNA here in red. And now about the process of translation. The ribosome scans the messenger RNA, and for each codon, three-letter codon, for each sequence of 3 letters, it has a molecule that is called a transfer RNA right here. It's different, separated from the ribosome. It just interacts with the ribosome. So this transfer RNA in one side of it recognizes these 3 letters that match the 3 letters on the messenger RNA, and in the other side of it carries an amino acid. So this is the interpreter, right? This is what interprets the 3-letter codons from the messenger RNA to amino acid sequence. It binds to the messenger RNA promoted by the ribosome, and then each amino acid is linked to one another to form the amino acid chain. So we saw the messenger RNA, the transfer RNA, and inside this gray sphere, which schematically represents the ribosome, you will find also ribosomal RNA. Some RNA molecules are embedded in it. And so these 3 roles can nicely be described only talking about the transcription process. Yes? Okay.
Where is the transfer produced, and where is the information stored, which triplets should be connected with which?
Okay, so outside the scope of this slide, obviously we have a mechanism of loading of tRNA with the correct amino acid, and we won't go into this right now, but definitely just know that, of course, every mistake in the loading of an amino acid in its not on its proper transfer RNA, and you will get defective protein. The interpreter has to carry with itself the correct tRNA that will be linked to a certain three-letter codon. So there's another process outside the scope of this class and this course actually that loads transfer RNAs with their correct amino acid. Was that the question? Okay.
But I just wondered how, like, how it is, how the information is stored, which triplet belongs to which amino acid, or is it some chemical reaction that is...
So this can be described as a key, and this key just each key has certain codon or the complement of the codon right here in this region, and it can, for chemical reasons, obviously, it can bind only to a certain amino acid, and that's what gives you. So really, any mutation in the tRNA molecule, mutations in tRNA molecules are usually lethal, because think about it, it would affect not only a single protein, but almost all proteins, which will have a certain amino acid. So each time, the wrong amino acid will be entered into a protein, and in these cases, usually the organism will be not developed properly and will not develop at all.
What? Does the RNA...
Ribosomal RNA.
Yeah, but what?
It has a structural role in the ribosome. So again, for many years, I think up to the late 90s even, RNA had only information, storage and transfer roles, and we know for many years right now already that RNA can also play a structural role in nature. Okay, this is the genetic code. And this is the most common representation of the genetic code, which is a triple code. Biologists don't like 3D, so this is a way to put three X's on the same graph or the same table. So in each side here you see the nucleotide in the RNA, and then you can match to it an amino acid. So for instance, what codes to what the codon U codes for, and the answer is phenylalanine. And UG leucine. So each triple code has some amino acid that it codes for. So this is what is called the genetic code. And a few things to note is first that it's degenerate to some extent. Degenerate, it means that, well, we have 64 combinations or possible combinations of triple codes, because we have three-letter code, and we have 4 letters that are used in this code, and we have 64 combinations, and we only have 20 amino acids. So some codons will code for the same amino acid, right? So this is the degeneration of the genetic code, and that's the reason why we can't take an amino acid sequence and decode it back again to nucleotide. And amino acids that are decoded by different codons, or I would say differently, different codons that code for the same amino acid, these four, for instance, serine, are called synonyms because they're like words that have the same meaning. So the word is the three-letter codon, and the meaning is the amino acid, and they have the same meaning, so we call it synonyms. And another thing to note in the genetic code is that some codons code for not for an amino acid, but rather for the termination of translation. For instance, UAA and UAG are what is called a stop codon. And there's a mnemonics to how remember these codons. So the translation is terminated when you are away, and there's you are gone. You are away, you are gone. There's another stop codon. Yeah, here it is, and
where is the R?
you are
You're gone away.
you're gonna go away or something like that. What's the question?
Okay. The A, I thought.
Reading the table is clear.
And
How to read the table?
some initiation codon.
Right, and the second thing is, of course, we have some initiation codons, which always codes for methionine. So every protein starts with a methionine.
So what is the left?
What is the left?
Left bottom valve in parentheses
So no, okay, there's some wobble. Never mind these
Oh, okay.
exceptions. Never mind these exceptions. Okay. So this is the genetic code, and we will end this chapter. We're going through very briefly the process, very schematically, the process of translation, mechanistically wise. So the ribosome scans the messenger RNA, and then it finds its start codon, the first amino acid to be entered into the P site is always in thionine, because that's the one that is matched to the start codon. And then at the second step, an additional transfer RNA goes in, carrying with it a second amino acid. The two are joined together by a peptide bond. This is exactly the reaction that the ribosome promotes. The two moved one step leftwards. Instead of the A and P sites, they will be now at the E and P sites. The first site, the entry site, is now released to the entry of the next amino acid, or of the next transfer RNA that will carry with it the next amino acid. Okay, and with that we'll finish. And because we have a couple more minutes, so let's try to see this movie. It doesn't always work. This is
to make a link with the presentation.
כן, אבל so what you'll see here is it's an animation that is built on a crystal that was deciphered by NEURONAL. So this time it's not a schematic, animation where you see Pac-Man with rectangles or circles or something. It's built on what we think at least is really out there in the nature. So you can almost get an intuition of what happens in there in the cell. And okay, she crystallized the ribosome, I think, 10 years ago or even a bit more. And the reason that they waited so long for the Nobel Prize to come is exactly the reason why we saw, as we saw in the NMDA receptor paper, is that they had to perform so many validations to say that the structure is indeed correct, is indeed biologically relevant. So what you see here is part of these validations. The ribosome can now be said to interact with several, okay, this is the binding between the two units of the ribosome. So now each atom here is really where it is in nature. And these are the transfer RNAs naked from the amino acid exit the binding site. And right here, this is the messenger RNA that is being that is read and reporting to us. The protein is synthesized. Where is the protein? I'm not sure right now. I think right here below. The elongated, this is the protein. It's being elongated. Each amino acid is linked to the correct one before it. Now, for instance, I'm not sure this movie has the validation. For instance, we know that some substances act on the ribosome of bacteria, and we didn't know how or why. We didn't have a mechanism. And today, with the structure of the ribosome, we do have. So, for instance, this is the stop codon. Clack, and it terminates the formation of the protein.
And it falls inside the ribosome?
It's a tRNA without an amino acid that just kind of, I think, blocks any additional catalysis of the ribosome and hence terminates the process. Okay, so it's not here, but if you go into this link, I think you'll find another movie that shows how the ribosome structurally interact with all kind of what's the medicine against bacteria? Antibiotics. With certain antibiotics. So you just see the molecule interact with the ribosome, and you get, just within a week, you get a mechanism for so many molecules for how they, why are they effective. And obviously, using this information, we can now make them much more effective or more specific and so on. So that was the ribosome. Okay, again, for next time, answer the question page for structure, for protein structure and function. That's that. Any questions?
You want us to write that down in the question?
No, as we did last time. We only, just come with the page and we'll do it together.
אם אני קראתי נכון, לפעמים קורא לי שני טקסטים, לא,